afternoon, everybody. I do want to start out by thanking Mary for the invitation for this plenary session. This has been a fantastic conference so far. John for the kind introduction, and to all of you for coming to the last two talks of midweek hump day, especially when the weather outside is so gorgeous. Since today's session is on solving today's challenges, what I decided to do was stick with our good old monoclonal antibodies or mAbs, because despite the recent trend of a lot of new therapeutic modalities that have come about in the last several years, and you're hearing about it at this conference as well, like cell therapy, gene therapy, newer molecules, monoclonal antibodies still continue to dominate our therapeutic landscapes, particularly the biotech landscape. In fact, if you look at the top 15 top-selling drugs from last year, and coming from Bristol-Myers Squibb, when I looked at the list, it gives me a tiny bit of pride to see that the top two drugs, the top two of the top 5 drugs are from BMS, but that apart, if you look at the top 15 drugs, 11 of them are biologics, and more importantly, 8 plus 2, so 10 of them are antibodies or Fc fusion proteins. Not only that, just last year alone, 13 new antibodies were granted approval in the US or EU over a wide range of therapeutic indications. And as we all know, there are several hundreds of them in various stages of clinical development by various companies. So kind of quoting our first plenary speaker from Monday, Yatin, antibodies are here to stay for quite some time. So now if we look at our journey from the first approved antibody to now, and it's almost like more than 3 decades, we have really come a long way in many respects. The first has been the evolution of the antibody technology. So the picture up there is of orthoclone, the first approved antibody, which was actually a mouse antibody. From that came chimeric, and then if you look at the antibodies today, they are most of them are human, at least if not, then they are humanized, and this has really helped this class of therapeutics take off in a big way. The other evolution has been in the types of the mAb constructs. So we have regular antibodies, antibody drug conjugates, fragments, bispecifics, and so forth. The third area, which has seen a lot of scientific development in the last several decades, has been the mAb processing technologies, which is obviously going to be the focus of my talk today. So that brings me to the overview of my presentation. I'll start with platform approaches because that is the industry gold standard for mAb processing, but I'll focus more on the latest trends that we are seeing across the industry. Next, I'll talk about some key processing and development challenges that are plaguing the industry today, and I'll give you some examples or case studies. And then I'll follow up with some evolving manufacturing trends, because whatever we are developing needs to go to manufacturing scale, and then I'll summarize my presentation. So starting off with platform approaches, a platform approach to process development for mAbs is really the established paradigm in the industry. And this is really possible, as we all know, because all antibodies have the defined Y structure, which makes it somewhat similar. Platforms, if you just take a look at the literature, as I was making this presentation, I really wanted to look at all the literature and all the work that has happened in the last 3 decades. Platforms really came about in the early 2000s, but they became it started to get increasingly adopted by all companies, big and small, because it minimizes development time and resources. It allows for multi-product harmonization at manufacturing scale, and it really simplifies raw material procurement. Given all of these advantages, the adoption of platform was a no-brainer. So let's look at some of the key trends of the mAb platform today, and moving sequentially from the upstream to the downstream process. For upstream, one of the big trends that we have all seen through the decades is this exponential rise in cell culture titer. When I started in the industry almost 20 years ago, if you got a few hundred mg per liter of protein, that was considered a very good titer. And today we are routinely talking about 10 grams per liter, and some companies are even aspiring for 15 to 20 grams per liter titer. At BMS, our processes have been on this growth curve as well, following the industry trend. So you can see from our first commercial molecule to where our recent pipeline molecules are, there's been a huge improvement in the cell culture titers, and this has been possible because of higher productive cell lines, improved cell culture media, and really understanding our upstream processes much better so that we know what levers we can pull to really improve the productivity. Talking about cell culture media, that has also undergone an evolution through the decades. We started with serum-based media, then came serum-free, then came addition of complex raw materials like hydrolysate, and now most companies have their own chemically defined medium. Some of the focus areas that people are working on is application of high throughput technologies for people who work in the upstream area. Using amber systems, the mini bioreactors is pretty much state of the art now. All companies are trying to design their own, as I said, custom defined media for specific applications and to increase their titers. People are using a lot of the omics tools now to understand upstream better, and then looking at better feed control strategies. So this is what, as you all know, what a typical antibody platform looks like. Upstream, downstream. Upstream, you have a few expansion stages going into the production bioreactor, some of the levers that I already talked about. On the right-hand side is the typical downstream process, protein A capture followed by viral inactivation, a few polishing steps, and filtration. I covered upstream, so let's look at the few unit operations in downstream. Starting with protein A, but this is the highly selective step that really forms the basis for the downstream platform, right? It is so selective that it makes the rest of the purification way more streamlined. Over the years, if you look at literature, there's been a good understanding of binding interactions on protein A with the mAb as well as the host cell protein, and this has led to better design of protein A media over the years. Some focus areas that companies are still trying to focus on is developing better and better wash strategies to maximize host cell protein and viral clearance, and that is really to maximize the benefit that protein A provide. And also, as some people who work in the downstream area know, protein A is a very expensive resin, so we are really trying to maximize the resin lifetime by more effective cleaning strategies, and also maximize binding capacity, which I'll talk about a little bit later. Along with protein A, over the years, there's actually been a lot of effort to come up with alternatives to protein A. And if you look at the literature, you can see that there has been some success in this regard, but the amount of development work that is needed to come up with a non-protein A alternative is a lot more, because it's very specific for the antibody. And in my mind, it kind of eats away from the advantages of a platform approach. So I feel protein A is here to stay, but what we can do is that try to, and this has also been talked about recently, is try to utilize the capacity of the protein A resin as best as we can. In the recent years, multi-column chromatography has really come up in a big way. It's a simplified form of using simulated moving bed, and there are many vendors offering products as well as companies looking into that. Also, the other thing that has happened is that the Repligen patent for protein A expired a few years back, so there are other vendors now trying to come up with cheaper versions of the protein A resin, apart from the big players like GE and so forth. So it is worth evaluating them side by side to see if it meets your needs. Moving on from protein A to the next step, which is the low pH viral inactivation step, which is very industry standard. But recently there has been a trend to add an additional or an alternative detergent inactivation step. The reason this is done is sometimes it's used as an additional step to get additional log clearance of viruses, and in some cases, if your antibody is low pH sensitive, it is prone to aggregation at low pH, which is often the case for bispecific antibodies. This might be an alternative viral inactivation strategy. Triton is the most commonly used agent for this, but if some of you know that there are environmental concerns, particularly in EU. So companies are continuously investing in alternative environment-friendly detergents for viral inactivation. There's literature around there, and there's a whole, if you look at it, there's a whole host of process patents by various companies trying to come up with their own proprietary eco-friendly alternative detergent. Moving on from the VI step to the polishing steps, since protein A is so specific, it really takes care of the majority of the purification factor. So the polishing steps are left with removing trace impurities like aggregates, residual host cell proteins, and hence the name polishing steps. It can be one or two based on the product specific need or the company strategy. There are various modes of chromatography which are used. Anion exchange is kind of the part of most mAb platforms because it gives good viral clearance and HCP clearance. If you need aggregate clearance, people have typically used HIC or CX, and some folks even use mix mode to get some unique selectivity. The other trend which has come up in a big way in the recent years is the need for high concentration drug substance to allow for subcutaneous administration. Again, Yatin in the keynote lecture on Monday mentioned about it from a drug product perspective, and this afternoon I saw that there was a drug product session on high concentration formulations. And the reason this is coming up is that from a patient experience perspective, a sub-Q administration is way better than an intravenous administration. And recently, some companies, many companies in fact, including BMS, have partnered with a new drug delivery technology called Enhance from Halozyme, which helps to open the subcutaneous tissue and allow for larger volume delivery of the product. But what it means, everybody has been talking on the drug product end, is what it means is that we also need high concentration drug substance to be ready to allow for this technology to move forward. So how do we get there? So the basic platform for mAb still remain the same, but now we have to put some additional effort into the UFDF step, the final concentration step, to get to the higher protein concentrations. And as you get to the higher protein concentrations, there are some challenges that one needs to be aware of. First is achieving the target excipient concentration in the UFDF step, which I'm going to talk about in a second. As you go to higher and higher protein concentrations, the viscosity of the solution increases. So there are pressure drop limitations that you need to take into account. You also, because you're at such high concentration, the yield of the process, the flush yield becomes important as well. And dealing with the sterile filtration of such a highly viscous solution is another area you need to look into. And obviously you need to keep in mind the stability considerations as you go to high concentration protein drug substance. In the interest of time, I will not have a chance to get into all of this, but what I wanted to give was a scientifically interesting area, which is the drift in the excipient concentration as you go to higher protein concentrations. What happens is this happens because of two main scientific phenomena which you can look in the literature. One is called the Donan effect, which as you go to higher protein concentrations, there is a preferential exclusion of charged species which are there in the formulation buffer. For example, histidine gets preferentially excluded, so the concentration in the drug substance drops. H+ ions gets preferentially excluded, for which the pH can get affected. And there is also a volume exclusion effect which impacts excipients like sucrose and uncharged excipients like sucrose and so forth. So when you do the final UFDF step, what you land up is that the excipient concentration in the final drug substance is different from what your formulation buffer is targeted for, and you really can't have that because the formulation concentration is kind of is designed with certain stability considerations. So what do we do during the processing to compensate for that? At BMS, we have actually come up with a comprehensive strategy. As you can see, this excipient drift is a function of protein concentration. So what we do is we find this excipient drift at the target protein concentration and compensate that amount in the diafiltration buffer so that the final concentration matches that of the formulation buffer. This empirical yet systematic approach has worked very well for our antibody platform and has really helped in our high concentration initiative. Apart from that, in parallel, we have also, through an academic collaboration, tried to develop a fundamental understanding of what this phenomenon is and build a predictive model. Moving from the trends, next I want to talk about some common processing challenges that is seen in the industry. And the first one is something that every upstream scientist needs to think about, that is matching product quality with change in upstream conditions, right? Since the whole bioreactor and the cells producing the antibody is a complex phenomenon, the most common issue that we see is changes in PQ attributes like acidic variance, glycosylation, and even in some cases unexpected product quality attributes like the color of the drug substance, aggregation propensity on stability when you make some upstream changes. What I want to show here is a case study where at BMS we did a very systematic study to understand what was causing an increase in acidic variance on changing a cell culture process. And what we found was that certain parameters, cell culture parameters like temperature and feed media, increased the oxidative stress in that environment, and that not only impacted cell culture performance like viability and titer, but it increased the supernatant peroxide concentration, which in turn led to some protein modifications like glycation, oxidation, deamidation, and that manifested itself in an increase in acidic variance. Understanding this at a fundamental level really helped our scientists to come up with the right lever. So we were able to come up with an additive in the cell culture media that helped to reduce this oxidative stress and meet the product quality requirements. And here's a reference if you're interested in greater detail. The next problem that has also been talked about in the industry a fair bit is low molecular weight formation due to antibody reduction. This typically happens during the recovery step, but it also manifests itself during the protein A stage. This is really a time and temperature dependent reaction, often difficult to predict, and often seen upon scale up, causing a lot of heartache and in some cases a loss of batch. Why this is a concern is that this reduction actually impacts the final product purity specifications, as you can see from the non-reduced CSTS results, and in some cases, so it affects the percent purity, which is a specification. In some cases, we have also seen that samples with more low molecular weight are prone to aggregation during stability, and there was a certain case where we saw that samples with low molecular weight bound to vitamin B12 from cell culture media, and actually that led to a pink coloration in drug substance. So it's one phenomenon happening way upstream that has a big repercussion down the process as well. So it is important to understand why this is happening so that we can come up with more holistic levers to come up with the best strategy. In the interest of time, I am not going to go into the detailed reaction, but what happens is that reducing enzymes are released by cell lysis during the harvest operation, resulting in disulfide bond formation. So we have to think of ways to prevent this reaction from happening. At BMS, we have taken a multi-pronged approach to kind of minimize this problem. If you look at the literature, and many companies have talked about this, most have focused on mitigation strategies. How do we prevent the reaction from happening? Since I said it's a time and a temperature dependent reaction, people have talked about chilling the cell culture and the clarified bulk, keeping aeration so that there is an oxidizing environment, processing the harvest as soon as possible so that that reaction doesn't happen, and in some cases even adding adding additives in the cell culture, oxidizing additives so that this reducing reaction cannot happen. And this is the first line of action for us as well. But we have taken it a step further and looked at how can we predict the low molecular formation risk earlier itself in the process. And you can do that by having the right assay. You can have like a free thiol assay, redox indicators, some probes, and even identification of biomarkers during the upstream process itself so that you can get an early read. And if you can predict the risk of low molecular weight formation, then you can apply all your mitigation strategies a lot better. And in some cases, if our prediction strategy and mitigation strategy didn't work and you still land up with low molecular weight, which actually happens, it actually happens in many manufacturing processes, then we have developed a rescue strategy to reoxidize them in the molecule. The next problem that I want to talk about, and it is important in the industry and we are talking about it, is PSA80 degradation. I saw there was a poster, and I think there are 2 talks on Friday as well on this topic. So that shows that this is an industry-wide problem. Polysorbates, PS80 or PS20, are widely used non-ionic surfactants during formulation because it helps to prevent protein aggregation as well as particle formation. Now, even though this is very commonly used, degradation of these polysorbates can occur through different mechanisms like oxidation and enzymatic hydrolysis. As you can see, we are showing a degradation profile. PS80 has 3 forms: monooleates, dioleates, trioleates, and it's the monooleates that degrades. Why this is a concern is that this, if it degrades in the drug substance, it would have an impact on stability because polysorbates are added for that stability purpose. There's been work in the literature trying to understand this and identify the culprits which cause this, and it has been found that the presence of trace amounts of host cell proteins, mainly the lipases in the drug substance, is the primary reason for this enzymatic hydrolysis. What we have done is that at BMS, to validate this fact, we have actually taken lipase inhibitors and added it into cell culture fluid and shown that the rate of PS80 degradation goes down significantly. The other thing is there has been a lot of work in the literature trying to identify which specific host cell protein is responsible for that, and one of the ones that has been talked about in the literature is PLBL2. And as you can see from the data here is that the rate of PS80 degradation is directly correlated to the PLBL2 concentration. But one interesting thing from this is you can see is that even at 0 PLBL2 concentration, there is still some PS80 degradation happening, which leads to suggest that it is not PLBL2 alone, but there are other host cell proteins, other lipases, which are present in very trace amounts, which are also responsible for this degradation. Some have been identified, but there are many others that, so it's a very complex mixture. Host cell proteins are a complex mixture that leads to this degradation reaction. Since adding expensive lipase inhibitors is not really a practical solution for a large-scale manufacturing, we really need to build effective purification strategies to remove these lipases, and that's what we have done. One thing I wanted to mention is another fact that complicates this issue is the assay which you are using to monitor this. So the way this reaction works, there's a substrate, and the enzyme acts as basically the lipase acts as the enzyme, and then you form oleic acid. So you can either monitor the decrease of the substrate or the increase of the reaction product, or indirectly the lipase itself. And there are various assays which I have listed. There is no right or wrong answer. Companies use different assays based on what sensitivity they want to get to and what the problem is. And some cases, even a few of them might be complementary. But what we have done is we have really developed a purification toolkit within the platform to minimize for PS80 degradation. We start with an effective wash on protein A to reduce the PLBL2 concentration as much as possible, and we have seen that has a significant improvement on PS80 degradation. Sometimes that's not enough because, as I said, there are other lipases that not all of them have been identified. So we have actually modified our first polishing step to decrease it further, and in some cases, even if that is not enough, we have our third level where we have replaced HIC, where we have replaced the second polishing step from CX to HIC, and that has helped in occasional cases. Moving along from the processing challenges, one thing I did want to briefly mention, which is an interesting side topic, is the impact of antibody subtype on processing properties. As you all know, most of the therapeutic antibodies approved are IgG1s, 2s, or 4s. There has been work in the literature trying to link the properties of antibody subclasses to processing properties, purification properties, and so forth. In the interest of time, I won't go into all of them, but wanted to give you an example where we saw interesting behaviors on CEX step based on whether it was an IgG1 or an IgG4. And this led to a very effective collaboration with research as well to the best design of the molecule. So at BMS, what we are trying to do is kind of come up with a tool called the developability tool. I think Yakhtin mentioned it from a drug product point of view, but what we see is that based on, this is a scatter plot, based on some simple properties like pI and hydrophobicity, the size of the circle means how easy it is to develop. So if you have a high pI molecule, low hydrophobic molecule, it is very easy to develop. On the other hand, the other side of the quadrant is the reverse way. So this is a good tool for us to interact with research and assign resources and timelines accordingly. The last section I want to talk about are evolving trends in manufacturing. As antibody processes have become more and more specific, more and more productive and sophisticated, there's been a trend in the industry to move from stainless steel 6-pack to more leaner, single-use modular facilities. So there's really been a drive towards smaller footprint, highly productive manufacturing facilities by using various elements of continuous manufacturing, something we like to call process intensification. The BMS site, the Devon site of BMS, some of you probably don't even know, our main biologics site, one of the key biologics site is only 40 miles west of here. Devon's is a very good example of this evolution. It started around 10-11 years ago as a commercial facility with stainless steel 6-pack. Soon we built based on the growing pipeline needs, we built our first single-use facility, but in 2 phases. The first phase was a traditional one, and the second phase was left open to allow for introduction of all the newer technologies, which I'm going to talk about, which are the various elements of continuous manufacturing. Now, there are various flavors of continuous manufacturing, and it's explained very well in this position paper by Charlie Cooney from MIT and Constantine. You can have a fully continuous process, end-to-end, and I don't believe anybody is there yet with mAbs, or you can have a hybrid process where you kind of pick and choose the different technologies that fits your needs based on the company strategy. So the technologies that we have adopted for upstream, we have looked at N-1 perfusion, doing perfusion for the N-1 bioreactor. We are still a fed batch company, but now we seed at a much higher seeding density because of the N-1 perfusion, and then we have a few advanced process control levers. In downstream, we have adopted continuous protein A capture by using multi-column chromatography. We have adopted automated viral inactivation and connected the polishing steps with what we call as poolless processing. And like many of the talks, we are looking into PAT tools as well. So this is what our intensified upstream process looks like. On the top is the traditional process. On the bottom is where we have converted the N-1 and N-2 into the perfusion mode while still leaving the batch bioreactor the same. What it does is that it decreases the time in the bioreactor and improves our cadence significantly. On the downstream side, as I said, we are using multi-column chromatography, and this really helps for a better resin utilization and drives the productivity. Automated VI for more robustness, and interconnected polishing operations for better streamlining. What does it all mean for our throughput? What you can see from this table is that if you look at the third row, a much smaller single-use facility now can match the projected output, the output of a stainless steel facility, a six-pack. And this is not just a development project anymore, not a fancy cool development project. We have successfully finished our first engineering run and is in the midst of our first GMP campaign as I speak. I know I am almost out of time, so this does bring me to my last slide. In summary, as we all know, monoclonal antibodies still continue to dominate the therapeutic landscape. Hopefully through all of this, even though platforms are the industry standard now, this is really not the death of science in the antibody field. Some processing challenges do exist still, and gaining a fundamental understanding and doing good science can really help to address these issues in a long-term manner and not have short-term band-aid fixes. Companies are constantly trying to drive value with process and facility intensification, as I gave you an example. And newer modalities, I didn't get a chance to speak about bispecifics and fragments today because I focused mainly on traditional mAbs, can be faced with additional challenges, but a lot of the learnings that we have learned through the antibody industry in the last several decades will apply there as well. Last but not the least, I really should thank the entire biologics department at BMS for all the work that I showed, but wanted to call out a few individuals for their direct help and support. And thanks to all of you for your attention. And with that, I'll be happy to answer any questions.